Normalization is an important step as far as any experiment is concerned, but as far as microarray is concerned, it is the most important component. So we have to normalize our data for different background noises. There might be some signal coming from the slide. Slide might have scratches, relative intensity or relative, this attachment to some specific color dyes that might affect some or that might have some biasness. So in order to remove those biasness or those experimental errors, the process of normalization is there. So it includes data cleaning and transformation. Then we have within array normalization. So for example, if you have a 2-die data, maybe within the same experiment, there might be some features, or now onwards we start using the word feature to represent those locations or spots on the array where we have our hybridization reactions or where we are observing some colored signals. So we do also have between array normalizations. If we have different arrays, multiple arrays within the same experiment, we might have one array performing differently than others, so we need to take into account. So in this way, we need to do this between array normalization. This data cleaning and transformation process involves the identification of flagged features. So some of the features, they are given some flag values. So flags are actually, when we get those output files, we observe some flags or some numbers associated with them. We call them as flagged. So if a feature is not flagged or a flagged value is 0, it means that feature is good. But we do have different codes and different for those different numbers present in that feature, in that flag. So we need to take into account or identify those features that are flagged. We need to do some background subtraction. So sometimes we have some the signal which is coming from the background. So we are interested in the foreground. Anything coming from the slide, maybe a scratch or a spot that disrupt our experimental results. So in order to avoid that thing, we need to subtract that background noise. We need to do log transformations since there is a big variation among our data. So in log transformation, we will see in the next couple of slides that we can bring those big variations into the data such that those genes with the small expression and those genes with very high expressions, you can put them within one graph or within one, you can say within one picture. So flag values, they are actually quality features. For example, we can have some bad features where we have high coefficient of variation, and then since we are getting some distributions here within each feature, we have a large number of probes. So within those probes are then attached to our target RNA, and in that case we are getting a distribution. So and then we need to compare across different spots. So in this way we can have a measure of coefficient of variation, so that is the CV. Negative features, we need to, sometimes we have more background signal as compared to foreground. So in that case, these features we can call them as negative features. So there can be flag representing that some feature is negative. Dark features are those that have weak foreground signal as compared to the background. So those different flag values, they can represent those bad features, negative features, or dark features. Good quality features have a flag value of 0, and flag features, they may be dealt as maybe we can remove them if we can afford, maybe we can refer back to regional image and correct it, or maybe we can develop some criteria how to deal with those flagged features. So background signal represents non-specific hybridization to the glass. So sometimes we have those hybridizations to the glass, so actually it's not the true signal. It's something that the target is attached to the glass. We can observe some natural fluorescence of the glass slide itself. As I said earlier, there might be some spots or scratches on the glass. So you should have some background noise always present in this. So that is the issue, or mainly the drawback within this microarray, which has been then improved by the new technologies when those next generation sequencing technologies they came up. So people start going after them because in microarray we have large number of issues like this, what we are seeing in this slide. We need to subtract background signal from the feature intensity. So when background is greater than intensity, foreground is minus background is less than 0, we have to remove those features. So in this way you will get a negative number. So these are your negative features. We can use the lowest signal intensity as the background minus subtracted intensity. So in the whole slide we can get different grades of intensities depending upon the expression, on the relative expression. So we can select the lowest signal intensity as the background minus subtracted intensity, and then we can use that number in order to minimize this background feature. We can also estimate the true feature intensity. Log2 scale is really helpful in these kind of experiments. So we can take the log2 of CY5 versus CY3. If you take the log2, basically it's log to the base 2. So everything represented in terms of the powers of 2. We can come up with this answer is log2 CY5 minus log2 CY3. So log ratio of 1 is actually in that case it's 2 raised to power 1. The answer is 2 raised to power 1. It means the top part of this equation, the numerator CY5 is higher, it's twofold higher than CY3. A log ratio of -2, that means 1 by 2 raised to power -2. So that is fourfold down regulated in CY5. So CY5 is actually 1 maybe in order to just understand in this example, whereas CY3 is 4. So in that case we will get the log answer as -2. A log of 0 is means that no differential expression. So CY5 and CY3 both have equal amounts of those intensities. Whenever you have this numerator equal to denominator, your answer is 1. So in fact 2 raised to power 0 in this, if you fit in log2 scale this 0, your answer is 1. So both the things are when they are equal, your answer is 1. So in that case there is no differential expression. So here is some interesting numbers. So we have numerator by denominator 1 by 16, 1 by 8, 1 by 4, 1 by 2, 0, 1, 2, 4, 8 and 16. So these are the numbers. If you correspond them with the log2 scale, 1 by 16 ratio is -4. So it's telling that denominator is actually 4 times higher than numerator. The thing on the top of this equation is 16 times less than the bottom one. 1 by 8, if that is the ratio, the answer is -3 and -2 and -1. So if you have 0, it's negative infinity and so on. A numerator which is 16-fold higher than denominator will give you a log2 value of 4. So rather than looking into these small numbers, for example 1 by 16 is 0.0000 something, rather than that we can look into this log2 scale, when here we have the whole numbers, where a negative sign will indicate that it is down regulated, mean the down part in the equation is more as compared to the top part, whereas a positive number indicates that the genes are up regulated, where the top part is greater than the bottom one. Here is another effect of this transformation. So we have a scatter plot of CY3 against CY5. So we can see the data points are quite scattered since there is a big range in the data. You look into the scale, down there the expressions range from 0 to 25,000 and you have big variations. And then on the CY5 you have 0 to it's 30,000. So when you transform that into log2 scale and then you plot it, you see that those data points are now quite close to one another. So they are like you can fit a regression line on them where you have an intercept 1 by 0.4 and then you have a slope that tells you how the values they change with respect to one another, how CY5 changes per each change in CY3. So that is 0.88. Intercept is the place where this line, this best fit linear regression line, the line we see over here, where it touches x-axis. So that is your intercept. If the intercept is 0, slope is equal to 1, then they are identically, those both channels, they are behaving identically. So CY5 and CY3. So whenever you have those CY3s, total CY3s, and whenever you have CY5s, you put them in this graph so it can give you an idea whether our results are okay. Is there any biasness? Is there some more, values going towards CY3 as compared to CY5? So in this way, you can check for those biasnesses for the attachment of those RNA to those these dyes. Here is another effect of this transformation. So you have some feature or some number or some value that is 1250, which is overrepresented. So 80% of the genes have this value or this number. So this kind of graph is called as a Parr law, where we have those very small individuals taking the big share, and then very large number of the ones which are having those very small representations. So here we have, for example, around 10% that have a value of this 3750. So in this way, when you take all those expression values and you plot it in this graph, and those raw values, they look like this. What if you normalize them in log2 scale, and then you take those, for example, here we take the values coming from red and we plotted them in log2 scale, so we see a pretty fair or a normal distribution. So here you can apply those t-tests or those other tests like z's, which assume that data should be a normal data. So the raw data that was not normal one, but here when you transformed it into log2, it kind of became a bell-shaped curve, and this way data is more normalized. So this is the effect of transformation on the data, guys. So we see that normalization in the experiments, they remove experimental errors and the biasness which arise, and this process that includes data cleaning and transformation. There is within array normalization and there is a between array normalization.